Genetic mutations are changes in the DNA sequence that can alter the structure or function of genes, potentially affecting an organism's traits. Here's an overview of the science behind genetic mutations. Types of mutations. Genetic mutations can be classified into several types. Point mutations. Single nucleotide changes, such as substitutions, replacing one nucleotide with another, insertions, adding one or more nucleotides, or deletions, removing one or more nucleotides. Chromosomal mutations. Changes in the structure or number of chromosomes, including deletions, duplications, inversions, and translocations. Repeat expansions. Repeated sequences of DNA expand abnormally, leading to genetic disorders like Huntington's disease and Fragile X syndrome. Occurrence and causes. Mutations can occur spontaneously during DNA replication, repair, or recombination processes. External factors like exposure to radiation, chemicals, mutagens, or viruses can increase mutation rates. Effects on genes and proteins. Mutations can have varying effects on genes and proteins. Silent mutations. Alter nucleotide sequences without changing amino acid sequences due to redundancy in the genetic code. Missense mutations. Change a single amino acid in a protein sequence, potentially altering its structure and function. Nonsense mutations. Introduce premature stop codons, leading to truncated proteins that may be non-functional. Frameshift mutations. Insertions or deletions that shift the reading frame of the genetic code, altering subsequent amino acid sequences. Inheritance patterns. Mutations can be inherited from parents or arise de novo, new, in offspring. Inherited mutations can be recessive, require 2 copies of the mutated gene for expression, or dominant. A single copy leads to expression. Genetic disorders. Many genetic disorders result from mutations that disrupt normal gene function. Examples include cystic fibrosis, sickle cell disease, and muscular dystrophy. The severity and symptoms of these disorders can vary based on the type and location of mutations. Research and diagnosis. Genetic mutations are studied extensively through molecular biology techniques, such as sequencing DNA, to understand their molecular mechanisms and implications for health and disease. Genetic testing helps diagnose genetic disorders and assess individual risk factors. Treatment and therapy. Advances in genetic research have led to therapies targeting specific genetic mutations, such as gene editing, CRISPR-Cas9, gene therapy, and personalized medicine approaches tailored to individual genetic profiles. Understanding the science behind genetic mutations is crucial for comprehending genetic diversity, disease mechanisms, and the development of therapeutic interventions aimed at treating genetic disorders and improving human health.